need to get information in space as well as in depth, and hopefully at some point in time as well. So I came up with the idea that we should call this Space Force, because there's an enormous advantage of being able to understand not only the genes that are being expressed in a cell, but where those cells are and where they're positioned relative to one another. And of course, I just couldn't resist calling it Space Force. I was going to call it Space Balls, so it sounds like Space Force was better. So our first speaker is going to be Fei Chen, and Peter will introduce him. And I did want to also mention our next speaker. Our talks are given on the first Monday of each month. So our next speaker is going to be Amjad Askari, who is a postdoctoral scholar in Mike Elowitz's lab. And as you may know, Michael is going to be giving a talk this Thursday at the Salk, although he's not going to be speaking on memoir, which is a way of doing lineage tracing at single cell transcriptomic depth while preserving positional information. That's going to be given by Amjad, who developed that technology. So with that, I'll welcome you all to this continuation of our series in single cell biology, Space Force. That's going to start with Fei Chen and continue with a series of really exciting speakers throughout the rest of the year. Peter, I'll turn it over to you.
Right, thanks, Jeff, and thanks, Jeff and Brian, for putting this together. Yeah, so it's really fantastic to kick off this series with Fei. I imagine most of you are already familiar with his work. So Fei is a core faculty member at the Broad Institute, an assistant professor at the Harvard Stem Cell and Regenerative Biology Department. He obtained his PhD in biological engineering from MIT, and actually only in 2016. Okay, so he's clearly on a stellar trajectory. He was a Schmidt Fellow at the Broad Institute from 2017 to 2020, and as I think you know, and why we invited him to kick off this series, Fei has already made really fantastic contributions to technology development and application of those technologies in the area of spatial transcriptomics and genomics. Okay, so Fei is one of the co-developers of Slide-seq, which allows near single-cell high-resolution transcriptomic profiling in situ of intact tissues, and has also developed methods for in situ genome sequencing to allow spatial imaging of genome organization in single cells. So, clearly some really important and exciting contributions to this area. And with that, I'll hand over to Fei and let him tell us more about it. Thanks very much for speaking, Fei.
Thank you. Thank you for that amazing introduction. Yeah, I'm really excited to kick off Space Force here today. And really today, yeah, I'll be sharing with you some of the tools and technologies that my lab is developing on spatial genomics. And I heard a little bit that, you guys are also interested in maybe having a future seminar series on temporal genomics. And I am also very interested in that, but I won't have too much time to go into that aspect. So, I want to start off my talk with, this video, and this is kind of like every cell in a developing Drosophila embryo from shortly after it's fertilized to when it's practically walking off the microscope. I think a lot of us are drawn to this video because it showcases the wonder, of biology, but it also showcases the immense spatial and temporal coordination that exists, right, in biological processes. And so while in Drosophila, this developmental process is fairly stereotyped, and we've had quite a lot of luck, not us, but in developmental biologists have had quite a lot of luck in labeling cells and molecules one at a time or a couple at a time to build up an understanding of how this process works. But really for mammalian biology and for disease processes, they're not really very stereotyped. And what we really need are tools that allow us to study these molecular mechanisms governing spatial temporal biology in that context. And so, I think you guys had quite a great seminar series on spatial single cell transcriptomics. And it goes kind of without mentioning that this has been a recent technological advance, has really revolutionized our ability to study like molecular mechanisms in cells. And that's because in single cell transcriptomics, you can take a tissue and break it down to its constituent cells, right? And you can sample maybe this transcriptome or maybe the epigenome of each cell. And this is really kind of powerful because it's comprehensive. It lets you look at the cell types and states and the molecular programs, and also maybe how those molecular programs change in the context of like disease or development or other biological phenomena. But I think like maybe as was alluded to already in the introduction, fundamentally we have to remember that what we're doing in these processes is we're taking complex tissues which have inherent structure, right? And we're dissociating them into like this slurry of single cells. And in that process, you lose this context for cell-cell communication, or maybe cell-cell interactions and differential expression as a function of where cells are localized in the tissue. And so we'd really like to know not only the states of cells, but maybe how they relate in relation to the tissue and how those states are an emergent phenomenon from the organization. Separately, I think as was touched on a little bit, cells in tissues are pretty dynamic, and they might migrate, they differentiate. And so we kind of lose that temporal context when we dissociate as well. We get this snapshot. And that's something that we would also kind of need new tools for in the field. So, today I'll really talk to you about approaches to allow us to look at, take the unbiased measurements of genomics. And by unbiased, I mean like unbiased measurements of molecules, which are usually DNA and RNA, and place that within the context of tissue architecture and structure. And this is really kind of driven by a couple of needs and motivations when we set out to develop these tools. One is just that, and I'm sure you all appreciate, the recent developments in single cell have enabled us to collect, quite large atlases of cell types and states, but we have no idea, for example, how these cells are interacting or where they're placed in the tissue. For example, this is a couple 100,000 cells from just a very small volume of the mouse bed nucleus. And this is, and what we really like to know are the interactions and relationships and communications between these cells. But at the moment, we have really good ways of collecting these high dimensional data sets, but not very many great ways of placing those cells in context. And I think the second is something that we all intuit, which is that we need ways to connect pathology, to tissue. We've had pathology for a long time, and just from simple H&E staining, we can recognize pathological regions and the differences between maybe malignant and benign, or maybe disease and non-disease tissues. But we have very few ways to connect those cell type and cellular architecture changes, pathological architecture changes with cell types and cellular programs and molecular mechanisms. And so being able to connect pathology to gene expression is something that we'd really be quite interested in. Beyond gene expression, maybe we'd also be interested in connecting pathology, for example, here in the case of amyloid beta plaques, to the surrounding cell types, and maybe how the gene expression in the surrounding cell types change, right? Okay, so, to address this loss of spatial context when performing single cell sequencing, we developed this tool called Slide-seq, which enables genome-wide expression profiling in tissues at about 10 micron resolution, which is about the size of single cells in mammalian tissues. And I want to mention that all of this work is a joint effort between my group and Evan McCosco's group, and we work quite closely, and we started our groups together at the Broad Institute around the same time. And so the big picture is that Slide-seq is that it effectively lets you do untargeted single cell sequencing and at the same time lets you know where those cells came from in your tissue. And so how do we do that? In Slide-seq, we deposit individually barcoded 10 micron polystyrene beads in a 2D monolayer on a surface. And each one of these beads has maybe a 10 million or so oligonucleotides on them. Each oligonucleotide has a PCR handle, what we call a spatial bead barcode, which is just that each bead has a clonal sequence that's unique to that bead, but different between beads. On each oligo, there's a unique molecular identifier to allow for digital counting of transcript expression, and then a poly-T tail, which allows for the capture of mRNA. And so we then deposit these beads in these 2D arrays. This is a cover slip that's 40 millimeters in diameter. And each one of these white circles is an array of about 100,000 beads in a 3 millimeter diameter. And so because the beads are deposited randomly, what we then do is we take a microscope and sequence the clonal barcodes for each bead in the array. I won't go into the details of exactly how we do this, but effectively, for each color on this microscope image, green, blue, red, and yellow, that corresponds to a different base, A, C, G, and T identity on one position in this barcode, and we go through and interrogate the different positions. And this is quite easy because the beads are quite large and quite fluorescent, and we can build up the sequence of colors and then the XY locations at each sequence location. We can do this up front, and so that one of the key strengths of actually Slide-seq is that once the arrays are made, it's very easy to then actually perform the experiment. We can take these arrays and take fresh frozen tissue and section a 10 micron section directly onto the array. Then you can take in a simple protocol, you can generate sequencing libraries, very similar to single cell sequencing, where you perform reverse transcription and then some amplification of the cDNA library. We then take that cDNA library and we sequence it on an Illumina sequencer. And so you get are results very similar to single cell sequencing, where you get for each bead the unique barcode that it's associated with. And you can imagine that is in a matrix on the rows where there's a unique barcode, unique bead on each element of the row. And then across the columns are the digital counts across each gene in the transcriptome. And now if you remember, we had kind of these colors which represented the different bases in the bead barcode as well as their XY locations. We can match them up. And now we have an XY location for each of these beads. And at each XY location, then that means you have the counts of the genes across the transcriptome. Okay, so what does this data look like then? Actually, Slide-seq has kind of a similar data structure as single cell data. And so naively, you can apply the same computational tools, such as dimensionality, reduction, and clustering. Here, this is actually one of our first Slide-seq experiments where we ran it on the mouse hippocampus. And on the very left, what we've done is we've clustered the Slide-seq beads and annotated the clusters with the cell type identities. These are the principal cell types of the hippocampus. In the middle, what we've done is actually, we've just taken the beads in the array and colored them by which cluster they belong to. And on the very right, you have what kind of this tissue looks like. For those of you who aren't familiar with the hippocampus, this is kind of the cytoarchitecture of the hippocampus. You can see that we reconstruct basically the tissue architecture, but beyond that, we can now place kind of like the principal cell types, like the granule cells, the hippocampus and the CA1, then this oligo layer separating the hippocampus from the cortex. And so the takeaway is that there's no microscope. We've actually turned the sequencer into a microscope. And in fact, there's these 10 micron pixels and they take images of genes across the transcriptome in a single experiment. And so I'll just quickly summarize kind of what collecting this high resolution transcriptome data allows for. One, it allows us to reconstruct the organization of tissues and connect it to molecular cell types. And because, you know, each bead is about the size of a cell, and we get untargeted genome-wide expression, we can kind of reconstruct the architecture of different tissues. For example, here you can see the reconstruction of the cerebellum with the principal cell types labeled, and you can see actually in the orange the Purkinje single cell layer of Purkinje neurons corresponding to this histology here. But beyond that, we can use it to discover new patterns of spatial variation within cell types. For example, in our manuscript, we used it to discover spatial gene expression domains in these Purkinje neurons in the cerebellum, and so and we've actually discovered, like in this just week, developed kind of an unsupervised algorithm that will identify within cell types what are spatially non-random gene expression patterns, and using that we define some subdomains of the hippocampus, of the of the cerebellar Purkinje neurons, and it also allows us to discover kind of and visualize spatial gene expression changes in pathology. For example, we previously demonstrated profiling the spatial temporal dynamics of a model of traumatic brain injury. And so this is a kind of a quick summary of some of the highlights from the first manuscript. For today, actually, I want to focus on some of the progress we've made since we originally published Slide-seq. And in particular, we focused on a couple of things. One is we focused on the ability of us to apply Slide-seq to a variety of different tissue types and organisms, both in human, early mouse development, as well as organoids, and in particular, pathological specimens, mainly human oncology specimens. And second is that we focused on the capture, molecular capture of Slide-seq, or how many unique molecules each bead captures on the array. And just through kind of a series of molecular biology bead synthesis improvements, we've actually improved the yield of Slide-seq about an order of magnitude, and to about 1,000 unique molecules per bead. And this yield is actually pretty similar to kind of first generation droplet technologies like DropSeq. And we actually anticipate, you know, that just if you compare DropSeq to like 10xb3, there's another factor of 3 in there that we could potentially get out. And there's no reason practically that, you know, an array-based capture on the surface should be any less efficient than in droplets. So we're anticipating some more production there. And lastly, we've actually open sourced the array production and computational pipelines, and this has all been deposited on GitHub and protocols.io. And so these improvements to the Slide-seq technology have enabled us to develop many more like analyses of the Slide-seq data. Right, here's just as a vignette. What we actually did was we profiled the developing mouse cortex at embryonic. This is actually embryonic day 15.5, and here you can actually see that here's a hemisphere, and in this box region is the developing cortex, where the cells develop from the left to the right, from the ventricular zone to kind of a structured cortex. And we've actually applied some single cell algorithms for pseudotime, or actually RNA velocity, where we can look at the ratio of intronic to exonic transcripts to look at the rate of gene expression change. But we've actually projected now this RNA velocity onto the spatial coordinates. And what you can do is then you can construct kind of a manifold, a spatial manifold on both the rate and the directionality of gene expression change in this developmental process. And then what we can do is we can identify genes which vary along this manifold. And we can then use that to learn kind of the relative enrichment of gene sets from human genetic studies of intellectual disability and autism. And how and the relative ordering those genes lay on to basically this spatial developmental manifold. And so we think that these kind of data sets and analyses will be very useful in understanding kind of in the future the spatial temporal localization of genetic risk variants in CNS tissues. So far, I've shown you kind of a lot of data on unsupervised clustering of data from Slide-seq. That's what I mean by unsupervised is that, you know, we just use standard single cell approaches where we act as if each bead is capturing RNA from a single cell. And that's mostly the case. We actually find that each bead captures RNA from a median of like 1.5 cells. And so there are definitely beads which are captured RNA from kind of a mixture of cells. And I can illustrate this kind of like pretty clearly with these two types of neurons, these two types of cells, the Bergmann glia and the Purkinje neuron, which occur kind of like interlaced in a single layer in the cerebellum. And if you look at kind of the unsupervised clustering assignments for beads which are assigned as Purkinjes or Bergmann, you have some singlets on the diagonal on the axes, but then in the middle you have beads which are called one or the other, but have some mixture of marker genes from both cell types. And really, if we want to like learn about spatial gene expression changes within cell types or maybe cell-cell interactions, it's really important not to assign these mixtures as one cell type or the other, but to know what combination of the cell types they are and deconvolve them. And so that's exactly kind of an algorithm that we recently developed for spatial transcriptomic data in general, including Slide-seq. We've shown also that it works on Visium or Merfish or other sorts of spatial transcriptomic technologies. And this is a supervised learning approach to decompose mixtures of cell types into single cells called robust cell typing decomposition. And the basic idea is that we might collect some spatial transcriptomic data with different pixels on it, and each pixel or point in the array have captured contributions from different cells. And you might also have a paired, and as I said, you know, it's actually very easy to generate paired single cell references. So you might have a paired single cell reference where you've already annotated the different cell types. And so then the idea is that you want to estimate in each one of these pixels in what proportion and what combination of cell types best fit the observed gene expression profile using kind of just like a reference-based probabilistic model. And at the end of the day, then you can take each position in the array and assign that to a mixture of cell types. And so, so what we've actually done we've done quite a lot of work to validate this computational approach with simulations with ground truth data sets. And what I mean by that is that we took actually single nucleus RNA-seq data and single cell RNA-seq data from the same tissue. And first, we showed that we can use this algorithm to classify singlets from one data type to another. So this is on the bottom. This is the cell type as assigned in a single cell RNA-seq data, and this is cell types as assigned in a single nucleus RNA-seq data. And we show that we can get basically very accurate like classification of cell types from one data type to another, with the exception of maybe some similar cell types. There's some bleed through. And then what we did is that we took cells from here and artificially constructed doublet pairs where we, by mixing, you know, one, for example, Bergmann cell and one Purkinje cell computationally. And then first we try to identify when cells are mixed into a doublet, and we find that we do decently well at that task. And then once we've identified the doublets, we can actually accurately decompose those mixtures into the fractional content of the constituent cell types. And so this is the ground truth, and this is the reconstruction in the decomposition. So what this allows us to do, if we go back to our example with these two intermixed cell types, is that we can use single cell RNA-seq reference to both assign the cell type and decompose it. So now we have kind of the predicted singlets on the axes, and then for the mixtures, we have actually like the proportion, and then we can decompose those proportions into the fractional content. And so what this allows us to do is it allows us to perform, not only does it allow us to like deconvolve the doublets, it also allows us to perform really high resolution cell type mapping, where in your reference, in your single cell reference, you often have the ability to finally subdivide, say, inhibitory neurons here as an example. This is 27 different subtypes of inhibitory neurons that live in the hippocampus. Here is the spatial data. The gray cells are actually like the excitatory neurons. And then these are the assignments for these 27 different cell types. And we have independent validation that these assignments are quite accurate. And here you can see that, for example, these are excitatory neuron clusters that form the different layers of the cortex. And so you can see this reconstruction of a cortical column. So in this sense, you're like kind of leveraging the higher, both like the higher sensitivity and also your ability to more finely grain cell types in the single cell reference. But beyond that, I think it's important to also like recognize that philosophically, there's been like a lot of work in trying to detect spatially varying genes from spatial transcriptomic data. And in general, these approaches have not been thinking about it from the abstraction layer of cell types. And if you think about it, the principal component of gene expression variation in the transcriptome is like cell types. And cell types are not spatially randomly distributed in the transcriptome. And so if you want to look for genes which have like interesting patterns in your tissue, the first things that you're going to find are cell type markers. But what we actually are interested in is not like discovering cell type markers through spatial methods. We're interested in discovering kind of cell type specific spatial variation within tissues. For example, here are several genes which are smoothly varying in CA3 cell type. This is an excitatory neuron cell type within the spatial context. And beyond that, we'd actually want to find out. So that's how gene expression is changing within a cell type as a function of tissue organization. But I think just as interesting of a question is gene expression change as a function of who your neighbors are. And so here we did a simple test where we looked at astrocytic gene expression, so like gene expression within astrocytes as a function of their neighbors. And so these are actually genes which have differential expression in astrocytes when their neighbors are, for example, excitatory neurons. And like, for example, like SLC6A11 is a GABA transporter, and this is actually known to be a gene associated with perisynaptic astrocytes. But in general, I mean, this is just one example, but in general, we want to condition on cell types to actually like deconvolve the contributions of cellular environments on cell states. And that's like the thing that like the most exciting thing to me about spatial data. Okay, so kind of a quick summary of this first section on Slide-seq is that, I've shown you how we've developed some experimental and computational tools for high resolution spatial transcriptomics. One thing is that we've really increased the yield in Slide-seq V2. We've open sourced the array production. And also one thing is that for these arrays, we've really scaled our ability to construct them. And so we're starting to offer kind of arrays to folks through a core facility at the Broad. And that's internal right now, but it should be opening up quite soon to external collaborators. And I've also shown you how we can leverage single cell atlases to assign cell types to spatial data. And I think, this is where really next level of analyses really begin to take off, where we're going to be able to look at spatial differential expression within cell types and cell-cell interactions. And these are kind of really the tools computational side tools that we're really excited to develop. And at the same time, we're collecting some tissue atlases. One, just as a platform for developing these computational algorithms, and two, as resources for the community. And so we've been working on a atlas for the mouse brain and some developing mouse atlases. And so that's kind of exciting. So hopefully we can share some of that data going forward. So for now, for the last maybe, let's see, 15 minutes of the talk, I want to transition and discuss some other work at a different spatial resolution, but and a different modality of biomolecule, but still the same fundamental problem, which is like, how do we address recovery of both the context of sequence and the context of space, right? And this is work that we developed on in situ genome sequencing. It's actually the first approach to directly sequence the genome within cells and tissues. And this is really work led by 3 graduate students over the last couple of years between my lab, Ed Boyden's lab, and Jason Buenos's lab. And so again, like as I briefly touched on in the beginning, but imaging approaches are actually really powerful because they give you this really high spatial context. For example, like these are like dendrites, and you can look at like RNA within single dendrites, right? But sequencing approaches in general allow you to look at like sequence context, like for example, where on the genome a transcription factor binds, right? But you lose kind of like the spatial context that you can get from imaging. And actually, this has been well recognized in the field. There's been a couple of approaches that have tried to bridge that. We've had Hi-C, which is an approach to kind of reconstruct the organization of the genome through like statistical interactions between neighboring pieces of DNA. Now, this is challenging to do in single cells, but also it is a statistical reconstruction, so it's not an actual direct visualization of the architecture. And at the other side, there's been really quite a lot of brilliant advances in DNA fish-based imaging, where you can kind of start tracing out the chromosome. In these sorts of approaches in general, they've been focused on really high resolution tracing of single chromosomes, but also you lose like the sequence context, like what is like de novo mutations or SNPs or maybe translocations. So you lose the sequence context. This is because it's a hybridization based approach. And so we're in general interested in when we set out and how can we sequence and image DNA in the same single cell, kind of like somewhere at the intersection of Hi-C and imaging. And so how does this approach work? It's kind of, we can take cells and normally, in Illumina-based sequencing, you might extract the DNA, you might amplify it and put it on that sequencer, and then you might align it to the genome. Here in situ DNA sequencing, we fix the DNA. So we fix the cells with just standard paraformaldehyde. We construct sequencing libraries directly in situ. By that I mean we amplify genomic loci and we use fluorescence-based sequencing similar to Illumina directly within the cells, and then we align it to the genome, but now we also know where those reads are in X and Y and Z. And so I'll just quickly go over how we do this, and this has really been quite a lot of like amazing like tech dev on the front of the graduate students solve a couple of key technological questions. So in situ sequencing, genome sequencing, we take fixed cells, and we actually use TN5 transposase adapters to paste in sequence directly into the fixed genome. And this can either be accessibility sensitive or not. And to make it not accessibility sensitive, you actually just have to denature the histones a little bit, maybe with SDS or HCl. And this is something that people do quite often for DNA fish anyway. So we took a standard DNA fish protocol and we found that you lose the accessibility sensitivity after you apply that and you put in TNF5 transposase. So now you paste in some sequencing adapters into your genome. Then we ligate these hairpins onto the ends of the sequencing adapters. That circularizes them and allowing us to do situ doing rolling circle amplification. And what that does is it generates clonal amplicons, where each amplicon is like some amplified piece of your genome. And inside of each of these amplicons are these repeats, where it's the insert of your genomic DNA, and then some priming sites. And kind of importantly, we included in each one of these hairpins a unique molecular identifier. And that's actually important because it's rather difficult to do long, and by long, I mean greater than like 15 to 20 bases in situ. And I'm sure all of us who have done sequencing a lot are used to, even in short read sequencing on the Illumina sequencer, something like paired end 150 bases, right? And but we actually kind of use a similar trick as what we're doing in Slide-seq. We can sequence this UMI. Actually, we sequence it 18 bases. And so here, you know, it's one amplicon. You can actually see, you can actually call this basically by your eye. This is like, you know, we did a lot of optimization on actually the sequencing chemistry and microscopy to generate such high quality in situ sequencing reads. But you can take this UMI, you sequence it in situ, you can recover the amplicons and just PCR them back out, put them on an Illumina sequencer, and do paired end short read sequencing. By short read, I mean paired end 200 bases. Now you can sequence for SNPs or mutations or other aspects of sequence context. And now you match, and at the same time, you sequence that unique molecular identifier on the Illumina sequencer as well. And now you matched it up, this UMI on the Illumina and in situ, and now you have the XYZ position of your reads. And for example, in this single cell here, you can see the reads colored by chromosome, and you can immediately see that you can resolve like chromosome territories, kind of like the 0th order organization of the genome. And so you know, in our paper, we performed some studies in PGP1 fibroblast cell culture and mouse early embryos. For the interest of time, let me just talk a little bit about what we did in the embryos, which is that you know, we were pretty interested in profiling the genome organization of these early embryos because there's a growing body of work, obviously, which suggests that the chromatin architecture is both dynamic and changing rapidly in these early stages of development, and especially during like zygotic genome activation. But at the end of the day, you know, these embryos are really not accessible to existing techniques. You know, single cell high-C can only be performed on zygotes by microdissection of the pronuclei, and it doesn't really scale to 2-cell or 4-cell stages. And also these embryos are quite large actually when you get to the 4-cell stage for DNA fish. And so you know, we thought this was kind of like a pretty unique and interesting opportunity for in situ genome sequencing. And the other thing that's really cool is that, you know, within a single 4-cell embryo, we can actually look at how the nuclear architecture vary within, you know, daughter or sister cells or cousin cells of the same embryo, which is particularly cool. And so we actually then perform from the study measuring the genome structure at 3 distinct stages of development at the zygote shown here, and then 2-cell and early 4-cell embryos. And here is the median reads per cell. And you could actually see that it goes down as you go up through the stages. And that's actually because like the number of reads that we get per genome is entirely dependent on the genome volume. And it turns out in these early embryos, there's a little bit of a conservation of volume. And so we actually get more data per nuclei in the early zygotes. We also collected at the same time as the genome sequencing data, the in situ sequencing data, we collected antibody staining, which is one of the beauties of actually doing kind of this in situ work, is that you can layer on kind of multi-omics. Here we labeled kind of lamin, DAPI, which allowed us to see like the pronuclei as well as, excuse me, nuclear density, and then SEMP-A staining. And so, for example, here's kind of one, when you put that all together, here's kind of like data from one pronuclei. This is a zygote and, sorry, one zygote with, and the maternal and paternal pronuclei are actually separated at this stage of the zygote. And you can see, basically you could assign like genomic reads, and at the same time, you can look at their distance to protein landmarks. And because we also get the sequence information, we can actually assign, this is actually kind of an inbred cross, so the SNP density wasn't that high, but we can still see, just based on the SNPs, the assignment of chromosomes to maternal versus paternal lineages, right? So here you see that the pronuclei are separated, so it's clear that one pronuclei has mostly maternal and one pronuclei has mostly paternal. Here you can actually see that these 3D architecture of these territories as well, along with the assignments. This is kind of a simple connect the dot model where we've just traced out the fold of the chromosome by connecting nearby loci. It turns out that you can use this. This is like the simplest thing. This is just visualization, but you can actually layer high C data on top of this to construct kind of like a constrained nuclear architecture model that has the folds. So what we did was we actually can use these stages to characterize kind of like the organization and the changes in the genome across these couple early stages. For example, in this is one cell of a 2-cell embryo, you can actually see that the haplotypes of the different chromosomes after that first cell division, they're on the same nuclei now, but they're actually completely polarized. There's no mixing, very little mixing between the paternal and maternal homologs. And actually all of the chromosomes are aligned on one axis. The centromeres over here and the telomeres on that side. These are mouse. The mouse chromosomes actually only have really one arm. The centromere is really on one end and the telomere is on the other. So that's, you know, that's why we're using the centromere to telomere as a measure of like one end of the chromosome to the other. And so you know, what we can do is then over developmental stages, we can kind of, for example, measure the mixing between different SNPs and calculate kind of the intermixing between the maternal and paternal haplotypes, as well as the distribution of mixing over time to look at different models of this genome mixing. And we can also look at kind of the polarization of basically of these chromosomes along this dimension. This is actually kind of a well-known thing called the rabble. Well, I don't know how well-known it is, but in cell culture has been observed, and this is called the rabble configuration. And we think that this is because the cell division times in these early embryos are really, really short. And this polarization of the centromere to telomere is actually a function of that. But if you basically look at kind of the alignment of centromeres and telomeres between neighboring chromosomes, they become very synchronized actually as soon as you go from the zygote to the 2 and 4 cell. So for the interest of time, maybe I'll just highlight just one really cool aspect of this data is that we can use the fact that we are sequencing the genome structure of cells within the same embryo to look at how genome structure changes or you know, it doesn't change as a function of like inheritance. So here, if you look at basically 2 embryos, this is one embryo up here and one 2-cell embryo down here, and this is just 2 sets of chromosomes highlighted. You can actually see that these two sets of chromosomes are actually like, just by eye, have similar positioning within the nucleus relative to each other and relative to their homologs. And since these are 2-cell embryos, these are by definition sisters. We can look at the 4-cell embryos actually, and we can look at now across all of the chromosomes inside the nucleus and look at the correlation of their positioning. So like how similar is one cell, the organization of one cell's genome, to the organization of another cell's genome. And in a 4-cell embryo, there's a pair of sister cells and a pair of cousin cells. And so if we assign, if we look at the distribution of the two most correlated guys, they're very correlated. And they're similar to what you would see from the 2-cell embryos, as in like, basically like sister cells have very correlated genome structures in chromosome positioning. And then if you look at the cousin cells, which is like the least two correlated pairs, they actually are more correlated than just random pairs of cells between embryos, suggesting there's still inheritance of this genome structure across 2 cellular divisions. And this is, I think particularly maybe interesting as we think about like early genome activation and the really early choice of cell fate as it goes from 4-cell to 8-cell. That's when some of these things happen, and we might want to consider basically like how we looked at the positioning of the chromosome, but if you look at the folds of the chromosome, there's also some inheritance of genome structure at the folding scale. And so we're pretty interested in how basically like this inheritance of genome structure leads to changes maybe in like inheritance of fate in these early embryos. And so maybe I just want to summarize on the in situ genome sequencing side. We developed the technology that allows us, like for the first time, to sequence DNA sequence directly within cells and tissues. We think that this is, really powerful for looking at like the organization of genomes, and we did it in early mouse embryos, and we get about 1 megabase resolution on the chromosome and genomic structure. And that allows us to kind of make comparisons of genome structure within intact embryos, but also look at kind of like the changes in organization of genome structure as a function of these early developmental stages. There's a lot of other, uses potentially for the technology. Maybe, for example, like in tissues and such as tumors, you might be able to discover de novo changes in ploidy or translocations of chromosomes. We actually demonstrate that we can see that in the embryos. And maybe you can also imagine using this to do some like long range haplotyping because of like the ability of you to spatially localize nearby reads, and perhaps even assembly of de novo genomes with this approach. And so, I want to say that in summary, I told you a couple of different strategies we're using to measure like different types of genomic molecules in the context of tissues at like high resolution and high throughput Slide-seq, and at at cellular resolution and high throughput Slide-seq, and then at very high subcellular resolutions in situ sequencing. And so, as I said, my lab's also pretty interested in developing like synthetic biology recording tools, which I don't really have the time to go into today, but I'd be happy to talk about that offline. And so thank you very much, and I'm happy to take questions. But before I do, I just want to, thank all the folks who did this work with me. In particular, Evan and I have worked very closely on Slide-seq with the team of Sam, Bob, who led the original work, and now Dylan and Evan Murray have joined in. And then, as I said, Andrew, Paul, and Zach have really led the genome sequencing work. Thanks.
Great. Thanks very much, Fei. That's really, really fantastic. Really enjoyed it. Thanks. We have a few questions in the chat. So I'll start out by reading off some of them, and then maybe we can just open it up and people can ask direct. So first question is, so how does Slide-seq perform in, so this is from Danielle Engel. How does Slide-seq perform in tissues with high RNase levels, such as normal pancreas?
Yeah, that's a great question. I have to say that we've never tried pancreas. We've tried some other tissues where, you know, if you look at this, there's this chart with like RNase level as a function of, you know, tissue type. And the main takeaway of that chart is that the pancreas is like way off the chart. And there's other tissues that are, you know, high, much higher than brain, but not nearly at the level of pancreas. And, you know, those tissues like liver or kidney, like work fine. I'm not sure. I would be worried though about, you know, the extremely high RNase contents of the pancreas.
Right. And Jeff has a question. Jeff, do you want to ask direct?
Sure, thanks, Fei. So first, I just wanted to say that was really a beautiful talk, literally and figuratively. But, you know, I had a conversation with Evan McCosco, and I would really like to hear from you how Slide-seq v2 compares to other methods such as seqfish or merfish in terms of cost, read depth, and you know, precision of localization. And the other thing is, it seems to me that what's really limiting for implementation broadly are the availability of the pucks that you have with the beads on them. And you said that these are being made in the core at Broad, but first, when would others be able to access them? Or, you know, can other people make them simply using the protocols that you've made available in GitHub?
Right. Those are great questions. I guess that's 3 questions. Maybe I'll start backwards from the from the last one, which is that some groups are trying to make them with the protocol from GitHub, and I think we've helped a couple of them, and usually they tend to be microscopy cores, and one group has succeeded. I would say that you know, the core at the Broad that's making them, they're already just like basically making them for my lab and like basically everybody at the Broad. And so they should be ready to, you know, try to transition to outward collaborations very soon. And the question about the trade-off between merfish. So obviously merfish and seqfish and these sorts of approaches are, I mean, microscopy based, and they are very good in the sense that they allow you for a targeted set of genes, hundreds maybe, allow you to localize and count the mRNAs for those genes. Now they do require, you know, a decent amount of microscopy expertise to set up in your own group. And maybe those sorts of things will be commercialized. But that's one of the main things, is there's some upfront cost. And the second thing would be that you have to choose your targets versus an unbiased, untargeted genomics-based approach.
Well, seqfish Plus, according to Long Kai, goes full genome. He can do the full genome. It's
right.
Resolution.
Right. I understand. But I think that there's an inherent like trade-off if you want in the amount of time it takes to do that experiment.
Yes. Like
both the time and like capital cost to do that experiment over, you know, 100 genes. I think 100 or a couple hundred targets is a very feasible experiment with merfish, and then, you know, very few labs are doing a, you know, a whole genome-wide experiments, right? And so, so that's, I think, the main trade-off. And I do think that, to answer your question of availability, you know, there's going to be quite a lot of companies commercializing array-based capture. You know, people are looking into licensing Slide-seq, but I think 10x, you know, is going to come out with, has come out with Visium, and they're going to come out with, you know, some higher resolution versions of Visium as well. And I think that the commercialization is going to play a big role in people's ability to use the technologies.
Can I ask just one other question that had to do with your deconvolution method, where you're trying to separate, you know, potential doublets from singlets of different states, and you know, in cancer biology, cancer cells often transition or reprogram into different states, and they acquire hybrid states. So how do you distinguish something that you might call doublets from something that's a true hybrid state cell?
Right. I guess the question is whether you see those hybrid states in like a single cell reference. If you see those things in a sampling of single cells. The other thing I would say is that you don't have to do that sort of deconvolution with cell types. You can also do it with like factors like NMF or like, you know, cellular programs. And that may help with that particular type of definition. Right, because you can say that this cell is a mixture of these programs. Now, whether you think that that is a doublet or you think that that is a, you know, a real cell that has the mixture of those programs, that's kind of, you know, that probably has to be determined independently.
Okay, thanks, Fei. And a question from Ulis Uzkareli. You there, Ulis?
Can you hear me? Sorry.
Go ahead.
Okay, very nice talk. I'd like to ask, with this genome sequencing, what percentage of the total, let's say, mouse genome can you sequence? Or when you map, what percentage of the...
Right, that's a great question. Now, per cell, I think
Okay, I think we've lost Fei temporarily.
percentage of the mouse genome, but over all the cells, then you... Hello? Oh.
Go ahead. Hello? Maybe you want to, at least at my end, you cut out for a bit there.
Yeah,
maybe
I said my internet connection is unstable. Can you hear me?
Yeah, we can now. Yeah. Okay.
Apologies. Oh, I just said that in each single cell, the coverage is rather low, maybe like 0.01 or 0.1% or something at best. But across many cells, then obviously your genome, your coverage is then becomes like, you can cover the whole genome. Maybe not even that many cells, like.
And thank you very much. And how about the Slide-seq? What percentage of the, again, genome can you cover with mRNA transcripts from a single cell?
Similar to like drops, it's like 1,000 UMIs or so. So it's like similar to like a drop-seq experiment.
Okay, thank you very much.
That provides the right context.
And a question from Pallav Kasuri.
Yeah, hi. Yeah, wonderful talk, Fei, and beautiful technology. I really love the creativity. I have two questions. One about Slide-seq, some of the assumptions that go into the way you do this. So you identify where the beads are relative to each other, construct this like Voronoi diagram that basically segments the area into the different regions that correspond to the different beads. And then when you assign these transcripts, you base that on the assumption that each transcript goes to the closest bead. Is that correct?
Well, we actually don't do this Voronoi thing. Yes, we do make this assumption that each transcript goes to the, it's the opposite way around. We make the assumption that each bead, each sequenced transcript from the bead came from something nearby. Because like when we sequence it, we know which beads go with which transcripts, just like when you sequence like you know, because like you sequence the bead barcode and know what the transcript identity is.
Yeah. And so do you, I mean, okay, so to put it another way, for each bead, do you assume that the transcripts all came from a cell that is overlapping in space with that bead?
Yes, we do make that assumption.
Is that, how sure are you that that's true? That it's not
We
possible that
measured,
move?
we made some pretty careful measurements of diffusion, which I didn't show in this talk. That says that the diffusion length scales are pretty low, like less than the size of the bead, and so that's why we're basing that assumption.
Okay. Yeah. I was just wondering where that came from. That should be easy
Yeah.
to test. Yeah. Okay.
That's a great
And the
question.
second question is more sort of a curious exploration of possibilities. I'm wondering if, would it be possible if you did immunohistostaining, like antibody staining? But nowadays you have all these antibodies with oligos on them.
Yes.
Could you use this kind of approach and then release the oligo and use that as a combined proteomics slash transcriptomics approach?
Yeah,
the
I
same
think so. I think, you know, there are some folks who are in other types of technologies who are doing that already, and it just requires the molecular biology being worked out. But yeah, that's a good idea.
Yeah, because you can imagine like you have these antibodies with oligos. You could just make a duplex, and you just need to release it somehow.
Yeah, I think that 10x, there's like some work where some other folks have published a little bit on that.
Okay. Sounds like it's in the works.
Yeah.
Cool. Okay. Great. Well, thank you.
Thank you.
Any more questions for Fei?
Actually, I would just like to follow up on that.
Go ahead, Clark.
What's your association with 10x, and how involved are you with their product development?
Oh, I am not. I'm not developing the products. I don't have any disclosures with my affiliation with 10x. I mean, I think that in some ways we're like developing we're like kind of pioneering like what sorts of tools can be done and what sorts of analyses can be done, and you know, it would be great if they could commercialize some of the things so that more people have access to it.
Yeah. Okay.
But I have no, you know, financial or other conflict disclosure with.
I guess for us as sort of you know, potential consumers or users of your product of the 10x products, it would be good to know if they have sort of the Chen stamp of approval. That
Oh yeah, for sure. I mean, I think 10x does a great job. I think the main thing is that their arrays are a little bit low resolution right now. They're at like 50 microns, or I don't know how you want to count it. It's 50 micron, 50 micron spacing. So, but they are definitely working on a high resolution, like something similar to Slide-seq, that is slated to come out next year.
And where does the nanostring geomics fit in with this? That's kind of substantially lower resolution, isn't it?
Yeah, substantially lower.
Does that have any advantages?
I'm not, you know, I think the main advantage is that they've highly optimized those panels to work with FFPE. And there's a lot of clinicians who've really like bought in, and, you know, we collect a lot of data and the data looks great. So I personally don't have a lot of experience, but, you know, I think it's really valuable to get that like clinical, you know, perspective, right, on the technology. So
okay,
they've definitely collected a lot of like clinical data.
So if you've got archived FFPE sections, then the nanostring would be the way to go.
Yeah, I think so.
Okay.
At least currently, but yeah. Thank you.
Yeah. Any other questions for Fei? If not, then thanks very much, Fei. It's a really wonderful talk and great turnout. We were up to over 150 attendees, I think. So great job. Thank you.
The record. Thanks, Fei. Appreciate
Yeah,
I had fun.
yeah,
Have a great day.
yeah. Thanks. And next meeting is March the 5th, everybody.
it. No, April 5th. April 5th.
Oh, sorry, April 5th. Yeah. Thanks, Jeff. We'll let you know by email.
Bye.
Thanks all. Bye.